DNA replication S phase. Recall that DNA synthesis, or replication, occurs during the S phase of the cell cycle. DNA definition. DNA consists of 4 bases. 2 of them are pyrimidines and 2 are purines. A purine always pairs with a pyrimidine. Guanine always pairs with cytosine, while adenine always pairs with thymine. The specific pairing provides the basis for replication and transcription, as well as for the techniques used in molecular genetics and recombinant technology. The semi-conservative mechanism of DNA replication results in a double-stranded DNA that contains 1 newly synthesized strand and 1 parent strand. To carry out the various duties of replication, several enzymes and enzyme complexes work together in highly regulated fashion. An enzyme called helicase separates the strands by breaking the hydrogen bonds between complementary base pairs. The opening between the strands is called the replication bubble. Replication of both strands goes in each direction from the bubble, resulting in 2 replication forks, 1 at each end of the bubble. Opening the strands allows the other proteins, such as primase and DNA polymerase, to begin the replication process. It is important to remember that DNA polymerase can only synthesize in one direction, the 5' to 3' direction. Because of this 5' to 3' direction of DNA polymerase, there are differences in how the leading strand and lagging strands are synthesized and how the enzymes work in each. Looking at just one half of the replication bubble, the right half, we can see that when the 3' to 5' strand is used as a template, synthesis of the new strand can be carried out continuously in its 5' to 3' direction. This is the direction of the orange arrow. However, the synthesis of the lagging strand is more difficult. Because the fork is opening in the rightward direction, but the synthesis must occur in the leftward direction, the lagging strand must be done in sections. A short section is completed in the right to left direction. The bubble opens further, the replication proteins jump ahead to the right to begin a new strand in the left direction, and so on, as the bubble continues to open. Several enzymes and other proteins are important in this process. Of special interest is that, even though so many enzymes must function cooperatively, the DNA is still replicated at 500 nucleotides per second in the prokaryote E. coli. There are relatively few errors as well. Replication is slower in eukaryotes, but they may have many sites of replication operating at one time. Often, crossing over is presented as random accidents that just happen to fix themselves. In reality, it is a highly regulated process. The holiday model has been altered somewhat since it was first proposed, but it does provide an explanation for how different combinations of crossovers may occur. These crossovers add to the genetic variability characteristic of sexual reproduction.